Well, thank you for the opportunity to present at this great TMT symposium. I'm happy to share our research about achieving higher measurement throughput with TMT-based single-cell proteomics. And if I could, we'll start off with the disclaimers. And if I could summarize these, I would say bottom line is do not sue thermo, and do not diagnose diseases based on what I present because I'm not that kind of doctor. All right. So with that, let's get going. Okay, so we are interested in analyzing single cells. It's kind of there's a shift underway in the biochemical analysis community in general. A lot of research is moving from bulk scale analysis down to single cell analysis. And the analogy I like to use for kind of the motivation for doing this is taking a smart speaker. In this case, this is, I think, an Amazon Echo device or any complicated electronic device. And imagine if you wanted to make another one of these, you would want to know what the chemical constituents of this smart speaker were, how much rubber and plastic and silicon and metals is present there. But imagine if the first thing that you did was take this and put it into a blender, and you get this pile of dust here. You analyze that, and that will give you plenty of material to measure. But what you're measuring is an average of the constituents kind of distributed. So you get an average and you lose all information regarding the spatial organization of those materials and a little bit of information on like trace materials might just get averaged out below the detection limits and be hard to see. So single cell analysis is more like taking a smart speaker and measuring each component individually. So that should provide a little bit more of a nuanced understanding of the biological system by enabling the original spatial organization to be tracked. Makes it easier to identify, say, if one little component was highly concentrated in a certain metal or whatever, you can identify that more readily than if that were spread throughout. But of course, if we're doing single cell analysis, we've just made each sample much smaller and we've made a whole lot more samples. So that leads to great challenges in terms of measurement sensitivity and measurement throughput. And so our efforts in this area really started making progress in about 2016, where we recognized that sample preparation strategies that were developed for bulk scale analyses are not necessarily appropriate for single cell, because you can see if we're homogenizing a biological tissue and then performing a bunch of chemical and enzymatic processing steps on that, and then transferring to another reaction vessel, purifying, things like that, we're going to lose some sample along the way. And if we start with a bulk scale sample, we'll lose a little bit, but we'll still have plenty to put into the liquid chromatography systems auto sampler. But if we start with a single cell and put it through this same workflow, we may lose all of the material and have nothing left to analyze. So in the early days, I was fortunate to work with Ying Zhu, who had a lot of expertise from his prior research developing nanopipetting systems. So using this essentially, you can imagine, a three-axis stage that has a very fine syringe pump that can pick up and dispense nanoliters of reagents and do the prep that way. So by doing this, we working in a small volume, we can minimize the surface contact of those proteins, minimize the adsorptive sites where proteins can get lost, and we keep the concentration of those proteins relatively higher by doing this. So it's essentially a simpler prep and a smaller volume prep that we did. And we called this workflow nanopots, which stands for nano droplet processing in one pot for trace samples. And you can see the whole workflow. We're dispensing reagents, incubating a sample, dispensing more reagents, and so kind of stepping through all within these 200 nanoliter wells that are microfabricated on a glass slide. And this really did solve a big problem, moving us towards single cell proteomics, because which really illustrates that sample prep was the bottleneck, because we were able to reduce our sample input with this original work from 5,000 cells down to 10 cells, and we actually increased our proteome coverage at the 10 cell level more than anybody had achieved, even with samples comprising 5,000 cells. And the reproducibility and label-free quantification all looked good. So to get to single cells and to make spatial measurements, first for single cells, we initially coupled this with fluorescence activated cell sorting. So because the nanopots chips, or these nanowells, they're just sitting on a surface, so we can stick them under a fax sorter and send 1 or 3 or 6 cells to each nanowell. And you can see proteome coverage was pretty modest to begin with, just 600 or 700 proteins per cell with this initial work, but that has since come up quite a bit, and I'll present on that. And to perform spatial proteome profiling, we've coupled the nanopots platform with laser microdissection, so we can just cut out regions of interest and sort of transfer those to nanowells, and then do the same nanopots workflow. We've applied this to a number of biological applications. This is one that we did early on, looking at the proteins involved in these hair cells within embryonic chickens at embryonic day 15, where these hair cells are at a range of different developmental states. And these are the hair cells involved in auditory signal transduction. So this is an area that Peter Barr Gillespie at Oregon Health and Science University really focuses on. But at the single cell level, looking at these hair cells at different stages of development, we could develop kind of trajectories for different proteins that are being, down-regulated in pseudo time, or upregulated, or kind of have a dip and then go back up. And we could find a probable source of actin for cytoskeleton formation. So that's just one thing where I think single cell proteomics has been useful. We've also teamed up with great collaborators, Ed and Amanda at Biogen, looking at motor neurons that are is from human post-mortem tissue, and these are from donors that had the neurodegenerative disorder amyotrophic lateral sclerosis, or ALS, versus donors, control donors. And because this is neurodegenerative disease, we were microdissecting out individual motor neurons from these spinal tissue sections and looking at differences in protein expression, not only between the control donors and the ALS donors, but based on the dysregulation and aggregation of this protein TDP43, you can see how it's nicely localized in control donor tissue, and even within a donor tissue of somebody that had ALS, some of the motor neurons have pretty normal looking physiology, at least in terms of TDP43. And then we can see that the pathology starts to progress. So we can find motor neurons that we think are at different stages of degradation there. And we can see what other proteins, by correlating according or stratifying according to that TDP43 pathology, we can see how other proteins are changing in their expression in sort of pseudo time or according to this progression. So this is some of the stuff that we've been using single cell proteomics for. And so if we fast forward to more technology development efforts now, for our for label-free workflow, and I know this is a TMT symposium, but I think talking about what we're doing with label-free sets the stage for what we're doing with TMT samples. So the current label-free workflow, we can use nanopots or other strategies or sample preparation to reduce sample losses. And we like to use ultra low flow liquid chromatography, typically lower flows than what commercial systems support. So like 20 or 50 nanoliters per minute. And the reason we do that is to maximize our ionization efficiency. And then we've explored different acquisition settings on our Orbitrap-based instrumentation to increase our proteome coverage. So for example, if you're doing label-free analysis, when you're isolating your sample to perform a MS-MS scan, so fragmenting and analyzing the products there, if we broaden out our acquisition, our isolation windows, we can actually perform a data-dependent acquisition analysis. But while we're accumulating that selected ion, we can also accumulate neighboring ions at the same time. And in this case, we can use the Proteome Discoverer 3.0 with the Chimeras search engine that's really made for disentangling and quantifying or identifying multiple peptides based on multiple product ions that are co-fragmented, so these chimeric spectra. So the wide window acquisition can be seen as a bit of a hybrid between data-dependent acquisition and data-independent acquisition, in that we're doing data-dependent acquisition, but we're using large DIA or size windows to do it. And in terms of how this works, this has been really effective in achieving deep proteome coverage. So you can see here, this is without match between runs based on just MS2, identifying proteins based on their MS2 spectra of the constituent peptides, versus also including identifications based on the intact precursor mass to charge value and the elution time, so using match between runs. So you can see that using nanopots and low flow separations and wide window acquisition, we're able to quantify 3,600 proteins from a 0.2 nanogram sized protein digest. So the digest roughly corresponding to the protein content of a single cell. And we can speed it up going from a 40-minute analysis down to a 20-minute elution, and we don't lose too much. So there are trade-offs, but we can go twice as fast and only lose around 10% of our proteome coverage. So that's a worthwhile trade-off for many things. We've applied the same workflow to actual single cells. So the previous slide was looking at digest. And again, you can see, and we've compared to data-dependent acquisition and data-independent acquisition. And this wide window approach seems to really help. We're still able to quantify more than 3,000 proteins per cell. And it's very easy to at least tease apart different completely different cultured cell types. But even, and I'm not showing that here, but even within one cell type, we can knock out a single gene and observe hundreds of proteins that are significantly differentially expressed as a result of that one perturbation. So that works well. And then we've made some additional efforts to simplify the workflow. So instead of one pot prep, but multiple steps, now we've evaluated different one-step mixtures. So we basically get a cell into a well, add a single reagent that contains trypsin and possibly a surfactant to aid with lysis and digestion. And what this is showing is the protease in this case is Promega's rapid trypsin lyse C that's been engineered to be stable at 70 degrees C. And they recommend just a 1 hour digestion. And we said, oh, well, we've been doing our cell lysis at 70 degrees for 1 hour. And this also recommends 70 degrees for 1 hour. So let's just combine all of that into a single step. So the 4 different mixtures that we evaluated were this protease plus a reducing agent, or the protease plus a non-ionic surfactant plus the reducing agent, protease alone, or protease. Basically, these two are getting rid of the reducing agent. And as long as we don't have that reducing agent in there, we get similar proteome coverage per cell. And so it looks like with or without the non-ionic surfactant works pretty well for sample prep. So now we have a one-step workflow. And then the most recent work that we've done is kind of going away from these these custom-made nano wells to just working within commercially available PCR 384 well plates, because we said, well, if we just use a small volume, we can just work at the bottom of one of these wells, and that has a similar surface. Basically, the bottom of a micro well is not much different from in terms of geometry from a nano well. And we use this HP single cell dispenser, which is now marketed as the TCAN Uno. So maybe go to TCAN if you want to purchase one of these to do both our cell and reagent dispensing. It's a nice low cost dispenser, and we feel like this provides a nice no compromise proteome coverage relative to nano wells, and it's in a more convenient format, very easy. So I feel like with this, at least for label-free proteomics, it should not be such a huge hurdle for a proteomics lab to become a single cell proteomics lab. I think this thing costs about $30,000. which is pretty cheap compared to like an LC system or the mass spectrometer. And you can just add one reagent. Within about an hour, you're ready to go and get just as good a proteome coverage as what we typically see with our nano wells. And of course, we can differentiate different cell types. We get a little bit lower proteome coverage for Jurkat cells relative to HeLa because they're smaller. And one other little comparison that we did was looking at the proteome coverage from our home packed ultra low flow LC columns versus a commercial nano LC column. This is a PEP map. The reason we chose that is because those are available in 50 micrometer inner diameters, so a little bit more miniaturized compared to some other vendors. And that does make a difference. All the pain we go through to operate at these ultra low flow rates does have an advantage in terms of the proteome coverage. So that's kind of setting some of the stage with our label-free single cell proteomics work. And while we've been really focusing on reducing these losses and maximizing our ionization efficiency, Nikolai Slavov and Bogdan Bugnik, they were developing the SCOPE MS and SCOPE 2 workflows for single cell proteomics. So this, while we were doing label free, they developed this TMT workflow, which I'm sure you're all familiar about. And Chris Rose, who just spoke before me, has also probably introduced this, but it's essentially a TMT workflow that also has one channel with much larger input, so that enables more stronger MS1 signal, easier to recognize and isolate, and have a stronger MS2 sequence peptide fragment spectra, and then the barcodes or the reporter ions can be used for quantification of those. So and Nikolai's group and others have continued to develop these workflows. We also recognize that the advances in terms of the SCOPE MS workflow and the analytical advances from miniaturizing sample prep and doing sensitive separations. These are orthogonal approaches that can be beneficially combined. So this is some work from 2019 where we essentially miniaturized the SCOPE MS workflow and did it in a nanopots chip. And then Ying Zhu, my co-developer of the nanopots workflow, developed this nested nanowells workflow. So within a nanopots chip, essentially what we were previously doing was prepping our samples here and then pooling them into another well. And what the nested nanopots workflow allows you to do is prepare your single cells and then just pick them all up and feed them into the auto sampler all at once. And then what the Slavov lab is calling the NPOP approach. It's essentially a nano droplet sample preparation on a hydrophobic slide. And so you can see these kind of miniaturized preps and TMT-based workflows work well together, and there's some sort of convergence happening there. So now if we look at TMT versus label-free for single-cell proteomics, we can see that for label-free, we have it's pretty simple sample prep. We can just sort cells into well plates. They all receive the same preparation, so you don't really have to think about different TMT labels going to different wells. and a one-step prep is now possible. In terms of quantification, we have 4 to 5 orders of magnitude of dynamic range. In terms of boosting strategies, we have match between runs and wide window acquisition that are very compatible with label-free workflows. And in terms of proteome coverage, we can achieve about more than 3,000 proteins per cell, even using, this is not using the Orbitrap Astral platform, but the latest and greatest. But this is a few years old quadrupole Orbitrap Explorus 480 workflow. We can already get to more than 3,000 proteins per cell. So that's pretty good. There's a lot of advantages to label free. One disadvantage is throughput, right? So in terms of throughput, it's very modest, right? What we're able to do, maybe 24 to 72 samples per day with this. Now, if we look at the label or TMT-based workflow, we're looking at multi-step sample preparation where we need to sort the cells, lyse and digest them, label them, quench the TMT reaction, and acidify that. Quantification, we need to be really careful to minimize batch effects and co-isolation and ratio compression that results from precursor co-isolation. In terms of boosting strategies, we can employ a carrier channel as the Slavov lab demonstrated. One thing that we've seen, most of the publications that employ TMT, even with a carrier channel, are limited to about 1,000 proteins per cell. And so that's a little bit of a limitation there. But where TMT really shines is throughput, right? It's not too much of a stretch to achieve hundreds of cells, to measure hundreds of cells per day. And that just becomes really nearly impossible with label-free. So I would say at this point, I wouldn't say either label-free or TMT workflows are going away anytime soon. But it looks like they each kind of have their pros and cons. And we would like to understand these and do a little better. So Chris Rose, I think, spoke before me, and he has already also talked about hyperplexing, and this is something that we saw some of the great work from the Gigi lab, hyperplexing at the bulk level, and we thought that even though the TMT based workflows have much higher throughput than label-free, it's still a lot lower throughput than, say, where what we can contemplate for RNA-seq kinds of strategies, right? So we're always looking to increase our throughput. So we thought we would evaluate hyperplexing. This is multiple modes of multiplexing that can be combined multiplicatively to achieve even higher throughput than what a standard TMT workflow allows, where we're limited to 18-plex reagents for TMT. If we TMT label combine with SILAC and do our TMT labeling of both the heavy and the light pairs, then we can potentially get to, say, 36-plex, right? In practice, or more. I know there are new lines of TMT coming out that are even higher. So with hyperplexing, combining orthogonal isotopic labeling and isobaric labeling, so say SILAC plus TMT, that's what we did, but you could also consider O18 labeling plus TMT or things like that to accomplish similar things. And then we get a different MS2 spectrum for the heavy and the light and the heavy versions of a given peptide. And then we look at the reporter ions for each of those for quantification. And so I think it's a attractive strategy. And if you've listened to Chris Rose's talk, you're also aware that there are some limitations here. So I wanted to show the HyperSCP workflow. So this is using the nested nanopots approach, but even denser, where we, with this kind of proof of concept work, we had each separated heavy and light and treated each one of those wells with different TMT, its own TMT reagents. So say TMT 127N would be here, and hitting one of the Silac light cells and one of the Silac heavy cells down here. Chris is doing it a little bit more elegantly now, just putting a heavy and a light cell into the same well and prepping them together, hitting them with the same TMT reagent for prep. So that minimizes the number of individual wells that you can have. And you can see here, looks like we're missing some cells, and that is correct. That's intentional to have place for, say, reference channels and negative control channels, things like that. So some of the initial results from the HyperSCP approach were that using TMT Pro, the Silac labeling efficiency was good, but our proteome coverage without a carrier or boost channel was kind of poor. It was not what we're used to seeing for label free. So you can see without Silac, we're getting about 500 and then down to about 400 proteins per cell when we're doing the so we're taking a hit by going from label free to TMT Pro labeling, and then we're taking another hit by adding the isotopic labeling here. And now that's without any sort of carrier proteome. If we do include a carrier proteome, when we go to with a 10 nanogram carrier proteome, you can see that goes up quite a bit. And then if we go from a 60-minute gradient to a 90-minute gradient, that goes up further, again, getting us to about 1,000 proteins per cell. So if we had a great LC system, which is sort of a separate project for us, but one that fed one gradient after another with no dead time in between, or actually this is with hyperplexing and even with those dead times in between. With a 90-minute gradient, we can get to about 200 cells per day. And with a 60-minute gradient, we can get to 300 cells per day. Now, if we eliminated all that dead time, 60-minute gradients with 28 plex single cells per analysis, that gets us to almost 700 cells per day. So pretty good in terms of throughput. Now, one thing that we observed, and Chris Rose has also observed this, is that often the software will pick the light version of a given peptide and miss the heavy version, or vice versa, and that leads to a lot of missing values. And even if we use you can see if we have a little bit more sample in our light channels than in our heavy channels, then we reduce our missing values in the light versus the heavy. And even if we use the pair picking algorithm built in with Thermo, our missing values are still about 40%. So fortunately, Chris, who is much better than we are at, accessing the API on the thermoinstrumentation and exploring alternate acquisition workflows and developing new algorithms. As you've seen previously in the previous talk, he is addressing this. And I think that's going to take this from being kind of an hyperplexing, from being maybe a nice early stage proof of concept to something that can really be practically used. But even as it was, you can see the variability for a given reporter ion is lower than with actual single cells than it is with our control channels. So that's reassuring that things are more consistent when we actually have a single cell in there. And then this is looking down here at 4 different cell types, and the overlaying regions of this PCA plot are the SILAC heavy and light versions of a given cell line. And so at least these are differentiated at the single cell level. So to summarize from the hyperplexing study, we can achieve a measurement throughput of around 300 cells per day, which would be more like 700 cells per day if we had a nano LC system. For example, we're working on these multi-column nano LC systems that can do all the overhead steps on one column while the other one is feeding peptides, and then just take turns to have 100% duty cycle. So we can get pretty high throughput with this, but we did see increased missing values due to missed SILAC heavy and light isotopic pairs. But as you saw from Chris Rose's talk, he's developing improved algorithm for minimizing these missing values. And another thing is, none of our work did we show, it was all with TMT Pro, and we never showed anything with more than about 1,000 proteins per cell. And we're really interested in increasing the depth of proteome coverage for single cell proteomics along with the throughput, because the deeper we can go, you know, those are just whole new classes of proteins that we can study that we would otherwise be blind to. So to look a little bit more closely at why our proteome coverage was not all that we wanted it to be, we started looking at TMT, the original TMT versus TMT Pro. And of course, TMT Pro has this proline-like reporter ion, so in early characterizations of TMT Pro, for example, this great paper from the Gigi lab from 2020, they also looked at, you know, are there some significant differences between TMT and TMT Pro? And so to look at this, they were just using the TMT 0 versus TMT Pro 0 and doing some comparisons that way. And what you can see is TMT 0 had a little bit higher proteome coverage than TMT Pro 0. But TMT Pro 0 had a little bit higher reporter ion signal to noise, which is proportional to signal, the way that's calculated in the software. So a little bit lower proteome coverage for TMT Pro and a little bit higher reporter ion intensity for TMT Pro, right? So that's what they saw. And so we have done similar studies at the single cell level, so or kind of much lower input level. So this is comparing our proteome coverage for label-free samples versus TMT 0 versus TMT Pro 0. And the sample in this case is the Pierce HeLa Digest, HeLa Protein Digest standards. And we're analyzing 13 nanograms, which is a weird amount, but the reason we selected that is that's the approximate amount of a TMT set that's analyzing single cells and including a 10 nanogram carrier channel. All right, so it's the same sample amount of 13 nanograms in each case. We're doing a 60-minute gradient at 50 nanoliters per minute for each of these, and using the Orbitrap Exploris 480. And what you can see here in gray, this is our label-free peptide coverage here for each of these sets. And we're looking at different collision energies here, expecting that some of these will have different optimal collision energies. And so we have label-free here, TMT 0, and TMT Pro 0. So what we're seeing is similar proteome coverage for TMT 0 versus label-free, but significantly reduced proteome coverage for TMT Pro 0. So why would we see such a difference between in TMT 0 versus TMT Pro 0 at the very low input level versus a more bulk scale experiment? And the reason I think for that is that we are much more kind of sensitivity limited than when we're at the bulk. We're more speed limited. So difference in fragmentation efficiency, things like that are going to be much more muted with higher input samples, where the instrument's already going as fast as it can. As we go towards the single cell level, if we're not fragmenting as efficiently, the sequencing cycle moves a lot slower. So we also have compared TMT 0 versus TMT Pro 0 and looked not at the proteome coverage here, but at the reporter ion intensity. And now the situation is reversed, okay? We're seeing much smaller reporter ion intensity across a range of collision energies for TMT 0, and then it's much higher for TMT Pro 0. And then just kind of poking around and looking at the most intense fragment ion intensity versus the MS1 ion intensity, seeing a lot more higher for TMT 0 versus TMT Pro 0. Okay, so it looks like we're really preferentially fragmenting, and this is a known thing. TMT Pro 0, the reporter ion resembles proline, and there's this kind of preferential fragmentation adjacent to a proline residue that's been observed, in proteomics for a long time. So what it looks like is TMT Pro 0 is really preferentially fragmenting and cleaving at the reporter ion, giving a much stronger reporter ion signal relative to TMT 0, but a much weaker peptide fragment ion signals relative to TMT 0. So there's kind of a balance there. And so we're looking at that and continuing to study that, because we really, since the whole main advantage of TMT-based analyses for single cell proteomics is for high throughput measurements, it's really beneficial in terms of throughput if we can effectively use the TMT Pro. So there's kind of more optimization studies underway. So just to summarize here, we've seen advances in sample preparation, separations, mass spectrometry acquisition and data analysis for single cells and other low input samples. Hyperplexing with TMT and SILAC together has given a kind of a demonstrated throughput of 300 cells per day, which can be increased to about 700 cells per day with a more efficient nano LC system. And studies are underway to increase proteome coverage for both TMT and TMT Pro based single cell proteomics. This time, I'd like to thank a wonderful group here at Brigham Young University. Yuran Liang, who did the hyperplexing work, is not pictured here because she graduated this past December and is now working at Genentech. But T. Trong led the wide window data dependent acquisition work, and the whole team contributes in a lot of ways. And I just wanted to let the group know that we have a single cell mass spectrometry conference going to be held from October 4th to 7th here at Brigham Young University. And so it's not too late to register, and it's not too late to submit a poster abstract. The oral program is pretty well set, but anyway, that should be a good place to learn more about single cell mass spectrometry, not just proteomics, but other ohms as well. And my fellow panelists, Chris Rose and Olga Vitek, will be there, and we're really looking forward to that. All right, thank you for your attention.